Dr. Katharine Barr. It is a great pleasure to have her here with us today. She is an assistant professor at the University of Pennsylvania and an infectious diseases physician, a clinical trialist and physician scientist who studied the basic mechanisms and translational impact of virus transmissional, pathogenesis, and persistence of HIV and other viruses. So she has dedicated her career to pursuing prevention and cure strategies for HIV-1 and AIDS, and now she directs the Center for AIDS Research Virology Core. So it is a great pleasure to have her here, and she's going to talk about the HIV reservoir. What do we know? Thank you, Katharine.
Okay, muchas gracias. Thank you so much for the invitation. It's wonderful to be here. I have just one complaint. It is totally unfair to make me speak after Dr. Paul Sachs, and I resent the country of Spain for that. But my brief time in La Coruña so far has been beautiful. Thank you for this opportunity, and it's a real honor to be here. So I was asked to speak about HIV reservoirs. What do we know? And so my goal today is to try to talk about some of the recent updates to our understanding, because the reservoir remains kind of a mystery to us still, but we are getting new ideas. So as Dr. Sachs just mentioned, antiretroviral therapy is obviously a miracle, and it has done so much for people with HIV. We know that HIV blocks virus replication and it enables a return to health. But if we stop those therapies, virus almost invariably returns. And this virus rebound is what reveals the presence of this persistent virus reservoir. There is a lot of controversy about exactly what persistent virus reservoirs are, but clearly this is the barrier to cure. These are the infected cells that we need to either eradicate or suppress in order for people to experience HIV remission, where they are not taking antiretrovirals and living without viremia, or to fully eradicate that virus. I think a reasonable definition for persistent reservoirs is a collection of long-lived cells that persist and evolve through antiretroviral therapy. These cells have integrated HIV provirus, and that makes them capable of reestablishing systemic virus replication upon treatment interruption. But what the field has really been exploring recently is how dynamic, how much change there is to this reservoir. And what I'm hoping, the way I'm going to frame the talk today is to talk about several steps of HIV infection and how they influence the reservoir and our understanding of the reservoir. So this is my plain language summary / outline for what I'm going to talk about today, going through the various stages of infection. In primary HIV, we know that the reservoir is established incredibly early, so within the first few days after exposure to the virus. We know that the use of antiretroviral therapy is actually what defines the reservoir, but it also, because of that return to health phenomenon, the immune system, the virus hijacks the immune system to be stabilized and even to expand the reservoir at the time of ART initiation. And then during ART suppression, this is where a huge amount of our research is conducted. And what we have learned is the CD4 T cell reservoir, which is the majority of the information we have. This reservoir changes, expands and contracts over time, and that seems to be a balance between immune clearance of those cells and cell proliferation. We also have increasing evidence that myeloid cell reservoirs are real and they need to be dealt with. So throughout the talk, I'm going to make vague references to cured strategies that sort of leverage the features of the reservoir that are identified through each of these time points, and hopefully inform our understanding of promising cure strategies that will get us there, maybe not in 5 years, but hopefully in the next period of time. So if we start with primary HIV, I think it's very clear that the reservoir is established extremely early. So these are data from a study published by Jason Whitney initially in 2014 and a follow-up study in 2018. And what they did was, using the non-human primate model, they did intrarectal inoculation of SIV to 4 rhesus macaques and started antiretroviral therapies at day 3. And what you can see here at day 3 is there's no detectable plasma viremia. Systemic virus replication has not yet happened. And so this is very, very early ART initiation. And yet, after 24 weeks of ART suppression, 4 out of 4 animals experienced virus rebound. So we know that that permanent, long-lived reservoir of infected cells is established within 3 days. Next, the group went on to delineate what happens in days 0, 1, 2, 3, as the reservoir is being established. And they started antiretrovirals at the same time as virus challenge, 1 day, 2 days, and 3 days after in each group of 5 macaques. And what they saw, unfortunately, is that even 1 day after virus challenge, the reservoir was established in some of these animals, 1 out of 5. Day 2, there's a little bit more frequency, and again, by day 3, 5 out of 5. So we know that very early on, we have a permanent infection of cells that will persist through antiretrovirals, and a very short window to prevent infection. And we have similar data from case reports in humans. So this is a study published by Tim Henrich from UCSF about an individual who was presenting for care to receive pre-exposure prophylaxis. And on the day they started pre-exposure prophylaxis, lab results came back and showed the beginning of acute infection. So we have viral loads of about 200 copies per mil. The virus went down on PrEP, and they have also started full antiretrovirals with 3 drugs. Virus was down within a week. Cells at this time had a small amount of RNA, but the person was started on the antiretrovirals and maintained suppression for 2 years on these antiretrovirals. Now, the team did extensive reservoir analyses. They did a leukapheresis and looked at literally billions of blood cells to see if there was any evidence of virus infection. They looked in the tissues, in the rectum, in the CSF to look for brain infection, in the inguinal lymph nodes, in the ileum and the rectum. And they were unable to find in any of these studies evidence of replication-competent virus. Then they did perhaps the most sensitive reservoir assay that we have with these humanized mice, the Mickey Mouse mice, as Dr. Sachs was talking about. And what they did was a humanized mouse virus outgrowth assay. So they took millions of cells from this participant, put them into the mice, and asked, does any virus grow from that? And what they saw was in 1 of the 10 mice, they had virus outgrowth. So virus was able to reestablish infection in this very humanized environment. So with this information of possible virus, but largely no replication-competent virus, the team and the participant had an informed consent process and decided to try a treatment interruption. And what they saw was this individual indeed maintained suppression for more than 200 days before virus rebound occurred, and then antiretrovirals were restarted and so that he could return to virus suppression. But again, what this shows is extremely early ART initiation, really at the first possible moment that a clinical diagnosis can be made, was not early enough to prevent reservoir seeding. So there are large benefits to very early antiretrovirals. The reservoir is much smaller, it's much less diverse, and we do preserve host immunity and prevent a lot of complications. But in adults in particular, we think very early ART is insufficient to cure HIV. Now, there's a possibility that that may be slightly different in pediatrics, and that's for several plausible explanations. One, we know that infants' T-cell immunity is a little bit different. They have much higher proportions of naive CD4 T-cells. They're not as likely to take infected cells and have those turn into long-lived memory cells. In addition, we know that at the time of birth, the babies and the mothers are accessing healthcare frequently, and so this is a time that we can intervene very closely after we think in utero infection has occurred. And so, of course, the classic case is the Mississippi baby, the Mississippi child, that was presented by Debbie Prasad initially in 2013. And this was a child that began antiretrovirals within 30 hours of birth, was maintained on antiretrovirals for 18 months until the family was lost to care. And then at 23 months, it was found that this child was off antiretrovirals but had maintained virus suppression. At that period of time, they did extensive reservoir analysis, and that child maintained a viremic state off antiretrovirals for more than 2 years before virus rebounded. So again, this is a very substantial but not complete reservoir reduction. But there are other case reports in children with early antiretrovirals after perinatal transmission showing durable virus suppression. So there's a case report of a South African boy who started antiretrovirals 2 months after birth, had extensive evaluations showing no replication competent virus, and has been off ART for now more than 10 years without virus return. In addition, there's a case report from a French girl where, again, perinatal transmission was followed by early antiretrovirals. This girl has had evidence of detectable virus at times, but has been maintained off antiretrovirals for many, many years with only small amounts of virus returning. And so because of this, there is efforts in the field to test whether in the setting of in utero infection, very early antiretrovirals may be able to reproduce these case reports. And again, the biological plausibility is that the CD4 T cells are less likely to be infected and revert to a long-lived memory state, so we may have a better chance at this opportunity, and we're going to try to build on these case reports of success. So the study that is being conducted right now is by the IMPACT Network. It's called the P1115 study, and it's looking to try to reproduce that clinical phenomenon of the Mississippi child. So at this point, the goal of this study is to enroll infants with in utero infection, start them on antiretrovirals within 48 hours, 2 days of birth, and then follow them for virus suppression and check for markers, biomarkers consistent with viral suppression. So this study opened in 2015 across 30 different sites in Africa, Asia, and the United States, or the Americas, and multiple children have been observed. The 2-year interim data were presented at CROI recently, and what we saw was 30% of the participants in the 2 cohorts that are essentially similar, 30% of these individuals maintained virus suppression on antiretrovirals for 2 years and also had labs consistent with possible virus control. So they had seroreverted to have no HIV-specific antibody responses, they had no detectable provirus, and they had normal CD4 T cell counts. And so the study is now working with the families and these children to consider whether a treatment interruption to test whether there's really been eradication or suppression of this virus is possible. And we'll look forward to these results in the near future. So let's go on to the next phase, and this is ART initiation. And I think this is actually one of the more hopeful areas in terms of cure strategies because we've learned some important new features of the reservoir about this time point. And what we've learned is the actual starting of antiretrovirals itself stabilizes and then leads to expansion of the reservoir that was circulating in cells near that time. So the study that's perhaps the most important in reinforcing this concept was published a few years ago by lead author Melissa Rose Abrahams from the CAPRISA cohort. And what this group did was they followed women who were enrolled in the CAPRISA cohort, identified during acute infection, followed with viremia until, this is an older study, until they met country guidelines to start antiretrovirals. Then they were suppressed on antiretrovirals and continued to be followed. The team sequenced plasma virus through this period and then sequenced the reservoir virus from outgrowth experiments later on. And what they showed in their study is if you look here longitudinally at the sequences in this rainbow color of red, yellow, green, all the way through to the time course of plasma viremia, the outgrowth viruses, shown here in pink, are highly enriched. They overlap with the viruses circulating at the very latest times just before antiretroviral initiation. And in fact, they're very highly enriched for this time point. If the viruses had been seeded into the reservoir at a rate based evenly over time, we would not see this. And if the reservoir viruses were seeded based on how many infected cells, we would see an enrichment for acute HIV when we know there's way more virus replication than there are during later time points. And so what we see here is there is this enrichment of the reservoir for viruses that were replicating at this particular time point. So what is the possible mechanism for this? It's possible that there's a different level of immune selection against these later viruses that are most likely to be escaped from immune pressures like antibodies and T cells. So that's a possible thing, that these virus-infected cells could better survive immune surveillance than earlier, more susceptible proviruses. But there's more data support that it's actually the immune stabilizing, the return to health features of antiretrovirals that leads these virus-infected cells to be more able to persist. The thought is that by introducing less inflammation and more normal immune function, an infected cell is more likely to be able to basically do what it's supposed to do, revert to a memory phenotype and last for a long period of time. And therefore, this return to health phenomenon allows those recently infected cells a better chance of persisting on ART. There's also the concept that there may be a dramatic T cell proliferation, expansion of CD4 T cells after ART initiation, as again, a part of this return to health phenomenon. So let's talk for a minute about clonal expansion, because I think it's an incredibly important part of how we understand the reservoir right now. So we know that long-lived memory T cells, the way they persist over time, whether they're infected or not, is by proliferating according to antigen, their cognate antigen stimulation, or just over time to maintain that population. But it wasn't until about 10 years ago that we first identified CD4 T cells that were infected with HIV that had clonally expanded. And so in 2014, this was first identified by looking and finding cells with identical proviral sequences and identical integration sites. And those two things happening by chance, are incredibly unlikely. And so we know that's because a virus-infected cell was then proliferating and carrying that same integration site and that same virus sequence on. Now, many, many studies have reproduced the fact that we see a lot of clonal expansion of HIV-positive CD4 T cells. And in fact, numerous studies have shown between 50 and 70% of all cells identified with virus are part of clonal lineages. But we know anytime we sample the reservoir, we are dramatically undersampling what is actually in the whole body. And so if we can extrapolate with modeling, we believe that more than 99% of all of our virus-infected cells are the product of expanded clonal lineages. Whether these are small or large depends, but they're all being expanded in order to last over time. And so what modeling by researchers from the University of Washington, Dan Reeves and Josh Schiffer, has suggested is that when we start antiretroviral therapy, there's a response and our body sort of returns to health. It recognizes that we have lost a lot of CD4 T cells that were killed during systemic virus replication. And therefore, we institute this massive proliferation of CD4 T cells. And that is more likely to drive expansion of these recently infected cells than more distantly infected cells. And so we know at 1 year of antiretroviral therapy, 99% of the cells, CD4 positive cells in our body that are infected, are in fact part of clonal populations. And we think that that represents the fact that there are small numbers of massive clones and massive numbers of small clones. But that may also be a reason why we have this expansion of that more recent virus reservoir. So how do we act on that and use interventions that can target this time when we think the reservoir is solidified and established? Well, there are multiple interventions that could be used at the time of ART initiation, but one has actually been tested, and that's broadly neutralizing antibodies. And the thought of why this would be an important strategy to use at this time point is twofold. So first of all, we know that antibodies, via their Fc effector function, can bind to infected cells, and help the immune system clear them. We think by using antibodies at the time of ART initiation, we may be able to enhance killing of those recently infected cells and perhaps block some of that cellular proliferation. The other extra property of broadly neutralizing antibodies as a part of the active immune system is the possibility that they could enhance HIV-specific host immune responses. And this is through a process called the vaccinal effect, which is actually well documented in cancer therapy, but has yet to be proven to actually happen in HIV. But the concept is, if you have antigen and antibody, in this case, virus and antibody binding together, these immune complexes engage with dendritic cells and have the potential to boost HIV-specific immune responses. And so we really want to use antibodies when there's enough virus around that we can have these immune complexes form in a large amount. But we know an Achilles heel of these antibodies is that if virus is replicating in the presence of them, they will quickly become resistant. And so the time of ART initiation may be an ideal time for this effect because virus is not replicating, but we know after antiretroviral start, there's several weeks where virus is still present in the system, enough that we can make immune complexes and hopefully boost the immune system through that process. So one study has been published last year led by Ole Sogard and his colleagues in Denmark. And that's the eClear study, which tested broadly neutralizing antibodies and a latency reversal agent at the time of ART initiation. And again, the hypothesis was that these BNAbs and the LRA could enhance restriction of the reservoir and improve immune responses through the vaccinal effect. So what they saw for all of their pre-specified aims was a signal of efficacy. So they saw within virus decline and virus-infected cells, faster declines in viremia and a greater decrease in infected cells in the people who got BNAbs. They also saw greater frequency and more potent HIV-specific CD8 T cell responses in the individuals who got antibodies with sensitive virus. And finally, in a subset of participants who underwent a treatment interruption, they saw in blue participants who received BNAbs and had sensitive baseline virus experienced greater levels of virus control. And so these positive signals have been very encouraging to the cure field, and there are currently many different trials right now looking at broadly neutralizing antibodies, as well as other interventions at the time of ART initiation, where we think we may be able to most effectively limit the reservoir and boost immune responses. Okay, so let's go on to the last time frame, and this is ART suppression. And I want to talk about reservoir dynamics as well as myeloid cell reservoirs. So for a long time, we've known when we measure the number of infected CD4 positive T cells, the reservoir is relatively stable and persists over time. So this is looking at virus outgrowth from CD4 T cells over years of antiretroviral therapy. And this study by the Silicano group was published in 2003, quite a long time ago. It showed a very slow decline, but a decline with a half-life of about 44 months. 10 years later, the group from UNC published a very similar study with the same method showing a similar rate of decline on more modern antiretrovirals. But we know that virus outgrowth assays underestimate the reservoir. We also know they take a ton of cells, they're very expensive, and it limits the number of participants and the number of time points we can feasibly study. And so recently, the intact proviral DNA assay was developed. And this assay is an attempt to make a more high throughput way of measuring for a surrogate or a reasonable estimate of replication competence by looking for intact virus. So by using a probe at the 3' and the 5' range of the provirus, we can get an estimation of intact virus compared to more total defective viruses. And what we've seen in the early studies is that this correlates nicely with virus outgrowth, but is about 10 to 100-fold higher. So it is probably an overestimation of the replication competent reservoir, but it correlates, so we think that it has some reasonable results. So because this assay requires fewer cells and is less expensive, it has been applied widely to larger cohorts. And what we've seen, in fact, is that here it is applied to the SCOPE cohort, looking at 81 participants with a median of about 7 years of antiretrovirals. And what they showed is that they had greater declines in intact provirus than they did in defective provirus. And again, an ACTG cohort of 40 participants showed greater declines in intact versus defective virus. And this suggests that the immune system is better able to recognize viruses with more intact virus, likely more able to produce virus produce proteins and show that to the immune system. So it suggests some immune activity over this time frame based on the declines in intact versus defective reservoirs. And this actually is exciting for people in the cure field because it suggests that our immune system is giving us something to work with. Our T cells, our antibodies, our various immune processes are able to slowly chip away at the reservoir, and that's something we can build on. But there's an important caveat to this work, and that it's largely based on the first 5 to 10 years of antiretrovirals. And so on a more sobering note, there was a study published just a couple months ago, again from the Silicano group, showing a more sobering picture of reservoir dynamics over time. And this is really a massive amount of work. They studied 42 participants with a mean of 22 years of antiretrovirals each. So we're talking about a very long period of antiretroviral therapy where these individuals were suppressed, and we can look at the reservoir in those T cells. And what they showed initially is that both looking at virus outgrowth and looking at intact virus measures, the reservoir appears to decline over this initial period, similar to what was seen in the previous 2 IPDA studies. But there's an inflection point about 7 years out. And after that inflection point, it appears that reservoir decline stops, and in some individuals, the reservoir actually starts to increase in size. And so that virus clearance is eclipsed by cellular proliferation. So we go from a slow decline to actually a very slow doubling time of about 23 years. And so this suggests that there are immune responses that are active and clearing, but the proliferative nature of these cells is really a daunting feature of the reservoir. So what are we going to do to enhance our ability to clear those cells and to shift the balance to clearance over proliferation? Well, there's multiple strategies looking to boost immune responses. There's also strategies looking to block cellular proliferation. The anti-proliferative studies are in early stages, but there is promise that this could be a novel approach. But I want to talk about one study that was well known to this audience, I'm sure, the ALEX-2 vaccine trial, which looked to try to enhance CD8 T cell responses. And the reason I mention this is because it's one of the few positive signals we have of an immune enhancing, a cellular immune boosting response that is associated with positive outcomes. So very quickly, I'm sure you're all aware of the HCI vaccine trial, but this group of DNA, MVA, and chimp ad vector delivered immunogens were given over a period of time. Immune responses were measured, and what they saw in the vaccinees was strong, broad, and polyfunctional CD4 and CD8 T cell responses. At treatment interruption, all of the participants experienced rebound, but if they compared the vaccinees and excluded beneficial HLE class 1 alleles, they saw that the vaccinees were more likely to have modest control and stay off antiretrovirals, and that the post-ATI viral loads correlated with the immune responses that they had detected earlier. So this trial is now a part of several combination studies combination approaches that are in study right now, and it'll be exciting to see how those results move forward. But I want to end talking about myeloid cell reservoirs because this has long been hanging over our head as a potential reservoir, but there hasn't been definitive proof for a long time. It is very challenging to prove. So I just want to mention that there's 2 types of myeloid cells that we think about with HIV persistence. One is yolk sac or embryonic cell-derived, fetal-derived macrophages. These are largely what drive our tissue reservoirs in our lungs, in our liver, etc. And the second type are bone marrow-derived monocytes that then become more short-lived monocyte-derived macrophages. Some of these can be a part of our tissue reservoirs, but they are less likely. And we tend to study these monocyte-derived macrophages in the lab because they're much easier to work with. We know they're very susceptible to HIV infection. And we know that tissue macrophages, when studied in immunohistochemistry and path specimens, are associated with having HIV DNA and RNA. But again, we have not definitively proved reservoirs. So I want to talk about 3 studies that have demonstrated stronger evidence that myeloid reservoirs exist. So this was a study from Janice Clevens and Lucio Gama from Johns Hopkins, and what they did was they adapted the same methods that we use to prove reservoirs in CD4 T cells to monocyte-derived macrophages from people on long-term suppressive ART. And what they showed when they compared here monocytes in squares and CD4 T cells is that there was detectable intact virus in the monocytes. These are short-lived cells, but these were people who had been on antiretroviral therapy for years to decades. They then went on and did virus outgrowth, and they showed lower but detectable virus outgrowth in the monocyte-derived macrophages, and these levels persisted over years between, months to years between visits. And so these are short-lived cells, but they come from these long-lived hemopoietic progenitor reservoirs within the bone marrow. And so it suggests that some of our long-lived bone marrow cells, in fact, are a long-lived reservoir for HIV that can then make these more short-lived cells in our blood. So I think we need to think about the bone marrow as a reservoir. But the real question has long been tissue macrophages, but they are incredibly difficult to study. So the first true demonstration of persistence in tissue macrophages came from the Bombsal group from France, where they looked at urethral macrophages from people undergoing gender reassignment surgery. So they had large amounts of tissue from which they could look for very rare macrophages. And what they found, again, was that in their macrophages, they were able to detect evidence of virus. When they looked in immunohistochemistry, they could find evidence of DNA and RNA in these tissues. And then they were able to do virus outgrowth and, in fact, grow virus from these tissue macrophages. The final studies to really prove these myeloid reservoirs have been conducted in the brain. So brain tissue is obviously very challenging to work with in humans, but the last gift, which is a recently developed cohort out of the University of California in San Diego, asked people with HIV altruistically to donate their bodies at death for a rapid autopsy. And using rapid autopsy samples from people's brains who were suppressed on antiretrovirals, teams from the University of North Carolina have done limiting dilution microglia culture. The cells look beautiful on immunohistochemistry. And then from these, they can derive brain microglial viruses that are distinct, compartmentalized, but related from the rest of the systemic reservoir. So I think this is one of the last nails in the coffin that we do have to consider myeloid reservoirs. And we need to think about how we're going to fashion our therapies to be more broad and not just focus on T cells. So in summary, I hope we've been able to talk about how HIV reservoirs are dynamic and influenced by each stage of disease. Primary HIV shows that the reservoir is established very early, and early ART will not by itself limit reservoir size or will not eradicate reservoirs in adults, but we do still believe it has some potential in pediatrics and is a component of a combination strategy in adults. ART initiation, we think, is key to stabilizing and expanding the reservoir, and there's optimism in the field about interventions at that time point to be able to block reservoir formation and boost immune responses. And finally, during ART suppression, we have realized this balance between immune clearance and cell proliferation needs to be tilted in the favor of immune clearance with better immune therapies, and we also have recognized that myeloid cell reservoirs really do exist. And so with that, I will say thank you very much, and I appreciate very much this opportunity.
Thank you, Dr. Barr, for this very interesting talk and for all the information that you provided about the reservoir. So I think that we can thank both speakers for this session. Thank you very much. Y muchas gracias a todos vosotros por vuestra atención. Creo que ha sido un comienzo estupendo para el Congreso y estoy segura de que el resto del Congreso vamos a disfrutar todos. Muchísimas gracias.